The rabies virus contains 5 viral proteins which are listed below. These proteins inhibit mechanisms within the innate immune system. The majority of transmission occurs from animals to humans. The virus is transmitted via saliva or through mucous membranes. Exposure typically occurs due to an animal bite. Only people at high risk of infection are vaccinated preventively. Post-exposure prophylaxis includes administration of vaccination and rabies immunoglobulin to provide virus-neutralizing antibodies to target the G protein of the virus. The innate immune system provides a much more prompt response than the adaptive immune system, which takes a few days due to clonal expansion. Within the innate immune system, PRRs recognize PAMPs, resulting in inflammation. Innate immunity hinders the spread of the rabies virus through antiviral cytokines and cellular responses. However, the rabies virus has developed mechanisms to essentially bypass the innate immune system. The type 1 interferon family of cytokines primarily organize the innate immune response to the rabies virus. Viral detection within the cytoplasm leads to upregulation of these cytokines. NF-kappa B triggers expression of antiviral cytokines. P43, which stabilizes the NF-kappa B complex, is inhibited by the matrix protein in the rabies virus. Intracellular replication of the rabies virus occurs via RIG1, which is a PRR that stimulates IRF3 phosphorylation and interferon transcription. The rabies virus nuclear protein inhibits recognition by RIG1. The rabies virus phosphoprotein prevents IRF3 phosphorylation, which hinders interferon transcription. Interferons bind to their receptors, which triggers phosphorylation of TYK2 and JAK1. This results in transcription of interferon-stimulated genes. The phosphoprotein in the rabies virus interferes with transcription, since this process is dependent on STAT1 and 2. The rabies virus also isolates developing viral particles in inclusion bodies, where viral RNA cannot be detected. Natural killer cells are the first cells detected after booster vaccination. They are responsible for early interferon gamma production after stimulation by IL-2 produced by antigen-specific T cells. NK cells serve as primary perforin producers, following early cytotoxic responses. Also, cytokine responses increase with elevated rabies antigen exposure.
The current rabies vaccine is categorized as a T-cell dependent vaccine, meaning that CD4 plus helper T cells are crucial in order to mount a sufficient antibody response to protect against the virus. To start, the inactivated virus originating from the vaccine is taken up by professional phagocytosing cells like dendritic cells. Then, peptide from the virus is presented on the surface along with the cell's MHC class II molecule complex. After the dendritic cell takes in the virus, the ingested viral fragments activate pathogen-associated molecular pathways. Once these are activated, the dendritic cell begins expressing CCR7, which binds to the chemokine ligand CCL21, directing the cell to migrate to the T cell zone within the lymph node. When both the dendritic cell and a naive T cell meet in the lymph node, there is an interaction between the T cell receptor, along with its corresponding CD4 plus co-receptor, and the MHC class II complex found on the antigen-presenting dendritic cell. In addition, the CD28 molecule on the T cell surface can then interact with B7 on the dendritic cell, as they are both being activated by the rabies virus. The CD4 plus T cells with affinity for the rabies virus, now fully activated, release IL-2 and undergo autocrine signaling to proliferate, undergoing many rounds of clonal expansion. Studies show significant CD4 proliferation occurring from day 3 post-vaccine onwards, with a peak seen around day 8. These peaks are before the B cell and antibody peaks, showing the T cell dependent nature of the immune response. Once proliferation has occurred, the many CD4 plus T cells can then go on to either activate macrophages or B cells. Those that migrate to the local site and activate macrophages can differentiate into subtype Th1 cells, releasing interferon gamma to cause other helper T cells to differentiate into Th1 as well, and causing macrophages to show enhanced antiviral behavior, strengthening the immune response at the site of the vaccine. Other CD4 helper T cells can become follicular helper T cells, migrate to the intersection of the T zone and the B zone, and through linked antigen recognition of the B cell's MHC class II complex, can activate these cells to begin producing anti-rabies antibodies. T cells other than CD4 cells, like CD8 and regulatory T cells, may play some role in the immune response, but their functions remain largely unknown. The most important type of T cell immunity-wise are the memory T cells. The central memory T cells remain in the lymphatic system and can quickly duplicate when the rabies pathogen is encountered, while effector memory T cells circulate throughout tissues and can immediately respond upon encountering rabies again.
The current rabies vaccine uses an inactivated form of the virus in order to create a B cell response that is dependent on the previously discussed helper T cell response. The B cells then differentiate in the germinal center into memory B cells or long-lived plasma cells, which are able to secrete the high-affinity post-switched antibodies. This can take from days to weeks to fully develop, and a full B cell response is really important for immunity. And the length of time required for this full immunity is why there's research being done to create a new vaccine. Virology is used as the main marker in studies of B-cell responses, which is accountable for the limited amount of research surrounding B-cell outcomes, especially in the rabies virus vaccine, but there are some studies that show us some of the B-cell response kinetics. A study on mice shows the B-cell response kinetics that allow us to determine whether or not this vaccine is effective. In this study, rabies-responsive CD5 plus and IgM plus B cells were detectable in mice before the vaccination was given. 7 days after the first dose, rabies-specific class switch CD5 plus G plus and IgA plus B cells were detectable. The antibodies that were produced after 7 days accounted for the primary B cell immune response. 14 days after the first dose, but 7 days after the second dose, IgM plus B cells responded by increasing 2 to 3-fold. However, 7 days after the final vaccination, the IgM plus numbers dropped back to the levels that were present previous to vaccination. For the Ig plus and IgA plus B cells that were rabies specific occurred after day 28, which is a week after the third vaccination, and this is the primary evidence of a delayed B cell response in the inactivated vaccine. Days after the primary vaccination was when the plasma cells and memory B cells peaked. The memory B cells were detectable from day 10 until the end of the study, but the plasma cells were only detectable from day 7 to 14. These peaks in B cell activation occurred after peaks in T cell responses, which supplies evidence of a T cell mediated B cell response. There are two problems with the inactivated rabies vaccine. The first one we previously talked about, and that is the time it takes to acquire full immunity. In the study we saw before, it took even up to 28 days to create a full B cell response. Another problem is that there is evidence that the pre-exposure vaccination can affect the efficacy of a post-exposure vaccination. And this is another reason why the vaccine isn't always used. A possible solution to this problem, using a mutated form of the virus that cannot replicate, is being currently researched. This form of vaccination shows evidence of T-cell independent B-cell responses through the production of virus-neutralizing antibodies in T-cell deficient mice. This allows for a more robust B-cell response when paired with T-cell dependent responses as well. Absence of T-cells, these B-cells were able to produce IgG and IgM. While IgM is not associated with protection against the RV infection itself, it may play a role in the overall immunity and vaccination efficacy. Antibodies produced during T-cell independent mechanisms help to provide early protection post-vaccination. However, these responses do not generate memory B cells, which is why the T-cell dependent B cell response is still important for immunity in this form of vaccination. T-cell dependent B cell response in this vaccine works the same way as it did in the previous vaccine. Evidence from this study shows that T-cell competent mice had higher RV specific antibodies than mice with no T-cells, showing that the use of both mechanisms of activating B cells creates a more robust B cell response.